So hello. So in this lecture video, we're going to be talking about, in this case, like the balance, sensation balance, okay, which is actually in this case mediated by the ear, and in this case, the ears actually mediates two distinct functions. So this area here, this region here, called which is by the vestibule as well as the semicircular canals, actually does like talks about balance, okay, and proprioception, which is position, your position based on where you are in space, and in this case the cochlea is actually the organ for hearing. So we're going to be talking about the cochlea soon, but both the sensation of balance and the sensation of hearing is actually in this case enters the brain through cranial nerve number 8, which is the vestibulocochlear nerve. And the reason why it's called vestibulocochlear nerve is because it innervates the vestibule and the cochlea. All right. Why do we have the vestibule and the semicircular ducts responsible for motion? Well, folks, in this case, when you're standing still and you're not doing anything, the primary region that detects that is your vestibule. And when you're moving, so this is called static equilibrium. And when you are moving, called dynamic equilibrium, the actual area that actually, in this case, mediates that senses that is actually your semicircular canals. Okay. We're going to talk about a little bit of a structure here. So this is actually how the structure, the sensory receptors are actually in your vestibule. Okay, so what happens is you actually have like this jello-like layer. Okay, it actually has a consistency like a jello. Okay, it has rocks called otoliths. Okay, not rocks, but little bony things. And what they do is they rest on these things called stereocilia. Okay, the reason why it's called stereocilia, folks, it has nothing to do with the stereo or like in this case sound. Stereo means space. All right, and what happens, folks, is when you move your head, the otolithic membrane moves. Okay, and when it moves, what happens, folks, is then there's a change. Okay, there's a change in the stereocilia, and if the stereocilia is open, what happens, folks, is the sensation then travels through cranial nerve 8, which is vestibulocochlear nerve. So when you're standing upright, the otolithic membrane isn't moving and the kinocilium is closed and there is no signal. So what happens is the standard nerve signal frequency is actually, well, let's just make up a number. Let's say it's 10, right? So that means 10 bursts of messages per second, okay? So that's frequency, right? So 10 shots per second, okay, going back to the, like in this case, to the brain, right? However, when your head is upright or down, so let's say again this is number 10, like, so this is 10 signals per second, right? So when your head is up or down, the frequency of the signals either increase or decrease. So if the baseline was 10, if your head is up, guess what? The number of signals per second goes up. So now we're going to say 20. If your head is down, the frequency of those signals go down, the frequency. So let's say 5. So again, I can make up any number. So let's say if the baseline is 100, and if I tell you that the new signal is 200 per second, you should know that that means to say that your head is upright. If I tell you that the signals per second is 50 instead of 100, then you should know that that is actually, in this case, your head is down. And that is because of the position of the stereocilia. When the stereocilia is, in this case, is in this position because of the otolithic membrane rubbing on it, okay, what happens, folks, is this actually opens up some receptors and more sodium actually enters and more ions actually enter. And when you go this way, you actually close, all right, the channel, so there's less ions moving in. And that is the reason why, and that is actually what we talk about static equilibrium. So how do you know whether your head is up or down or straight ahead? Well, it's about the baseline signal. If I tell you the baseline signal is 100, right, 100 signals per second, and I tell you that the signal for this person is 100 signals per second, guess what? Your head is straight up and down, right? If this person tilts their head back, their signals are going to go up higher than 100, okay? And again, this is the 100 number that I'm quoting or citing is actually just a very, very random number that I generated, okay? And then we have the semicircular canals, which then has this very important function of in dynamic equilibrium. This is actually how you can tell whether you're moving left, right, up, down. Okay, and there's no accident why you have three semicircular canals. And if you think about it in this way, you actually we live in three dimensions, right? X, Y, and Z. So guess what? You can think about the semicircular canals as actually having those same three dimensions as well. So one is about up and down motion. The other one is obviously left and right motion, and obviously in this case diagonal motion is actually in this case together, they all work together for that motion. All right, and it has a similar structure as the autolytic membrane or the stereocilia previously here, but what previously mentioned. But the only difference is it's the same thing. What happens is there's this gelatinous layer. When this gelatinous layer moves, because your head is moving, it induces a change on the stereocilia for dynamic equilibrium. Okay, so that is it. So here's an example, right? If your head is rotating horizontally, meaning to say in the what axis is that, folks? That's the x-axis. What happens is this is the semicircular canal. Okay, that's responsible. Then what happens is the endolymph, in this case the gelatin, moves. In this case the stereocilia is induced to actually take more ions in. All right, so that is basically it for static and dynamic equilibrium. Take home points here. Static equilibrium is actually integrated, or in this case sense, in the vestibule, and dynamic equilibrium, when you're moving, is actually integrated in the semicircular canals. Right? This is a specific pathway, and there's a lot of cranial nerves involved here, folks. And the reason why there's a lot of cranial nerves involved in terms of vestibular pathway is your body wants to stay upright. So when your head goes up, it has to sense that your feet is still on the ground. So that is why a lot of this pathway is actually connected with skeletal muscles. All right? And your eye, your ear together deduces your body position. All right? So this is one of the reasons why we get seasick, car sick. Okay, so predominantly what happens, folks, is there's a big discrepancy between the message that you're getting from this nerve and from this nerve as well. All right. These are actually, folks, these are not. I want you guys to understand that this is not the optic nerve. These are motor nerves, right? Oculomotor, trochlear. Okay. Trigeminal, if you want to think, and not actually not trigeminal, but so. All right, so that this is actually and what they do is, depending upon your head movement, then there's a message that actually gets sent to the eye to make sure that it actually can determine where your position is. All right, and based on that, obviously some skeletal muscle. This is actually not just again, remember, and again, on this this sensation is actually in this case partially integrated in the parietal lobe, and actually it actually is in the integrated in the parietal lobe. All right. So moving on, hearing. Hearing is also, in this case, sensed by the ear, obviously. Same thing, same architecture. You have, in this case, a kind of like an autolytic membrane. Okay, it's called the tectorial membrane, and you have, instead of being called stereocilia, it's actually called, in this case, inner hair cell. Okay, but you can call them stereocilia, kinocilia, right? So whatever you want, but they're the same architecture. Tectorial membrane moves, the tips open or closes, and what happens is there's actually an input of ions, and that ions then creates a signal to the actual, in this case, to specific areas of your brain. And I hope that you remember where hearing is integrated. It is actually integrated in your temporal lobe as well. All right. FYI, I forget to mention, the actual integration center for balance is actually in your cerebellum, okay, which is in the posterior and lower portion of your brain. Okay, so moving on, hearing. How does hearing work? We might as well talk about this whole architecture. You guys can read, but I'm gonna try to break this down for you. So you have your big ear here, the pinna. It looks like a trumpet for a reason, because what that happens is, folks, is it actually takes all the sound and traps the sound and concentrates it into your external acoustic meatus or external auditory canal. the eardrum then vibrates because of the sound, because sound is obviously in this case waves, okay? And this vibration then is actually propagated through the three auditory ossicles, malleus, incus, and stapes, okay? Malleus because it looks like a hammer, a mallet. Incus because it looks like an anvil, and stapes because it looks like a stirrup. So as this vibration travels through, what happens, folks, is that vibration, which is air vibration, is transferred into fluid vibrations. Okay? And this fluid vibrations then moves the tictorial membrane, and similarly what would happen with the otolithic membrane, when the tictorial membrane moves, folks, your hair cells then, the kinocilia in this case, right, the hair cells, what happens, opens up, and then they get more sodium in, and that creates the sensation that you're hearing something. Okay, quite important. Now, here's a very, very important thing. What happens, folks, is the consistency of, there's two types of fluid. There's actually perilymph and endolymph. Perilymph is more watery, and endolymph is more viscous. Okay, what happens, folks, is the reason why you have two different types of fluids is this is one of the ways that you can actually hear very faint noises, very faint, okay? And the reason why that is because all of this architecture is you're taking a very, very small sound wave, and what you're doing is you are creating lots of stimulus, okay? You're actually expanding that signal, okay? More important things for you to know. Loudness, right, which is measured in decibels, is based upon the vibration of this, in this case, this tympanic membrane, the eardrum. The louder the noise, the more vibration you get. The fainter the noise, or the low, right, the less vibration they get. Okay, louder, more. Less, obviously less. Pitch. Right, we know that we also hear different types of like pitches, okay? High pitches, folks, is actually in this case is actually triggered more on in this case on this area here, and low pitches is actually more in this case detected by this area. And the reason why this is there's a difference in stiffness of the base, in this case of the basilar membrane. If you want to think about it, this area is more wobbly, and this area is less wobbly, and that is the reason why this area actually can hear high pitches, and this area can actually hear low pitches, right? Because low pitches, right, what happens is low pitches has less energy, so it doesn't move this area, it only moves the wobbly area. Okay, high pitches, right, what happens is they're actually much more, in this case, much more energetic, so they actually move the more stiffer area. Now you might say, well, Abner, what does that mean to say that, well, everything should be wobbly? Well, it does, but in the case of the ear, it's the first area that wobbles that makes in this case, that actually makes a difference. All right, so again, waves, sound waves, vibration of the tympanic membrane goes through malleus, incus, stapes, goes through your perilymph, vibrates the endolymph. And FYI, there's something wrong with this is actually a typo, folks. This is actually not on this region. It actually is on this region here. Okay. And the tictorial membrane then vibrates depending on the region. If it's high pitches, this is the place that vibrates. If it's low pitches, it's this area that vibrates. And the hair cell then gets stimulated. And thus a signal then goes to the brain via cranial nerve number 8, which is vestibulocochlear nerve. Cochlear implants, what does it do? Well, folks, the only difference between cochlear implants is that you actually go through the brain, right, through the temporal bone, and what you actually do is you actually snake, and in this case a probe. And FYI, this is cochlea, folks, is actually circular. It actually looks like a snail. What happens, folks, is that actually deduces, so you actually have a receiver on the outside, and based on the receiver from the outside, if you have a cochlear implant, it stimulates specific regions of this, and therefore then it kind of like replaces the function of this whole of the cochlea as a whole.